Um, Hi everyone. Um, I'll be talking about focused ultrasound for gene therapy, and I've been working with Dr. Natasha Shifani. So, as I've had the privilege of working at the foundation for the past 2 summers, some of you may already know me, but I'm going into my final year of neuroscience at the University of Exeter in the UK with an interest in neurological treatments. My plan this summer is to focus primarily on cross-mediated gene delivery to the brain. So just a brief background on gene therapy. It's a process where we genetically modify cells to either restore cell function or to express certain receptors which affect cell function. So there are two administration methods. The first one on the left is ex vivo here, where cells are collected from a patient. They're genetically altered using lentivirus, and then they're reintroduced back into the body. And then we have in vivo on the right, where an injection of a viral vector such as adeno-associated virus, or AAV, containing nucleic acids is introduced straight into the body. So just a quick summary on blood-brain barrier opening. So the tight junctions between endothelial cells become wider as the microbubble oscillates, allowing entry of vectors containing genes or other therapeutics. And in the paper mentioned on the right here, it's been shown that pulsed low pressure focused ultrasound is ideal for stable oscillation, so stable blood-brain barrier opening for gene delivery. So I'm just going to go over some preclinical examples of using focused ultrasound to enhance gene therapy. So in the study on the top right here, lipid polymer hybrid nanoparticles, so LPH nanoparticles, were loaded in with small interfering RNA or siRNA. And they were used. So in this image, it shows 10 minutes after sonication, a 12-fold increase in the extravasation, or in other words, delivery of LPH in the FUS-treated region compared to the non-FUS region. And then in the study in the middle here, brain-penetrating nanoparticles, specifically luciferase plasmid-bearing brain-penetrating nanoparticles, were loaded with specialized plasmid DNA. So just looking at the two bottom images here, we can see a signal of 3.5 and 4.5 fold higher in FUS-treated brains. And then in the bottom images here, LPNs were also used, carrying specialized plasmids again, targeting something called MGMT, which is a drug-resistant gene to the cancer drug temozolomide. So these CRISPR-Cas9 plasmids target MGMT and they downregulate it. And that results in increased sensitivity of glioblastoma cells to TMZ. So just looking at after 21 days for control and post-FUS, we can see suppressed tumor growth already. So moving on to Huntington's, which is due to a CAG trinucleotide repeat expansion in the Huntington gene, which results in an abnormally expanded polyQ sequence. So it's been shown that the Huntington gene is actually suppressed by something called cholesterol conjugated siRNA, so CC siRNA. And we can see in the top study here that there's a dose-dependent correlation between decreases in the Huntington gene and the concentration of CC siRNA by actually up to 32%. And then in the bottom study, neurotropic factors were used. So for example, GDNF, plasmid DNA. So that showed reduced Huntington aggregates by up to 87% in the cortex and the striatum. So just looking at the beta tubulin here, which is the green fluorescence, that's showing increased neuron branching, which is increased neuron growth and viability, which is what we want. So moving on to Parkinson's disease, the study on the top right here shows a significant increase of GFP expression, which is an indicator of successful GDNF transduction in dopamine neurons, which will eventually recover normal secretion of dopamine. And the same thing here in the middle study, where dopaminergic neuron density is restored in lesion rats that received FUS gene delivery, actually up to a fivefold increase. And then the last study here, Intern was used, so NTN, encoded in the adeno-associated virus again, which I mentioned before. And this is immunofluorescent staining, just showing much more dopaminergic projections in green on the AAV plus positive side of the brain. So overall, with Parkinson's, we're looking for an amelioration of the nigrostriatal pathway in Parkinson's disease. So that's what I'm working on so far for the gene therapy mega document. And the plan is to compile most of that research into a review paper, as well as being involved in the gene therapy workshop. So I'm really excited to be involved with the foundation again, working with Dr. Shibani. So thank you all for having me.
Thank you, Juliette, and especially for joining in, given that you've just started your summer internship like 3 weeks ago or something. So kind of an interim update. Are there any questions for Juliette? If they're, I guess, forward-looking while she's starting her program. Chat. Check the chat. No. Okay. Well, thank you again, Juliette, for showing up.
Thank you.